Animal testing is usually considered controversial because of the suffering of the animals involved. But many scientists believe that its consequences for human health are equally controversial and deserve more attention. Europeans for Medical Progress Trust is a charity whose goal is to improve human health and protect patients by ensuring that all medical research is relevant to humans.
It's clear that there are serious problems with our current systems for making sure new medicines are safe and effective. For example, in March 2006, a clinical trial at London's Northwick Park Hospital made headlines around the world when 6 healthy young men were nearly killed by a new drug which was given to them after it had been shown to be safe in monkeys at 500 times the dose. The US Food and Drug Administration, the world's largest drug regulator, has complained that we're using 20th century tools to develop 21st century medicines. Fortunately, there are many new technologies and companies addressing this problem by focusing on the way that the human body responds to new drugs before actually testing them out on people in phase 1, 2 and 3 clinical trials. Randall Charlton is the founder and chief executive of Asterand, the world's leading human tissue company.
We operate a network of hospitals around the world in about 15 countries, and we collect under strict ethical conditions tiny amounts of human tissue backed by large amounts of data, all properly anonymized to protect patient and donor identity. I think it's self-evident that mice are not men, and in order to develop drugs with more confidence, wherever possible, one should use the human model, the species in which the drug is eventually going to be given.
It's important to remember that the pharmaceutical industry is developing drugs for use in humans, and for that reason a focus on human biology, I believe, is very important. Now, historically, the industry has relied very much on the use of animals, both isolated tissues and whole animals, but it's also had its problems. I've got many years' experience in the industry, and I worked for 30 years in a major pharmaceutical company where the use of animal tissues was the norm. And the thing that frustrated me there was the lack of predictivity of many of the data that we generated. And even at that time, I made as much use as I could of human tissue in trying to focus on what I thought was relevant.
An ingenious new technology that brings us closer to mimicking the whole human body in the safety of the laboratory is being pioneered by Hurel Corporation in California.
Clearly, there's a pressing need for better tools to accurately predict human response to potential therapeutics. Accurate prediction of human response is difficult, often unreliable, and invariably expensive. There is considerable concern whether animal studies can predict human risk precisely enough. To address these limitations, we have developed an interactive, cell-based microfluidic biochip. The Hurel, which is a condensation of the words human and relevant, is composed of a network of interconnected microfluidic compartments and channels, which replicate the organ and the blood system of the human body. In this example, there is a lung compartment as a target tissue for toxicity, a liver compartment, which is important for drug metabolism, a fat compartment, which is important for accumulating drugs and breakdown products, and an other tissues compartment, which replicates the holdup of circulating blood in other tissues of the body. The culture medium is a blood surrogate and recirculates in the Hurel system just as blood recirculates in the human body. The prototype biochips are roughly the size of a postage stamp and are fabricated in silicon using standard microfabrication techniques traditionally used in the microelectronics industry. But most importantly, the geometry of the chambers and channels are designed to mimic key aspects of the human body, such as the time taken for a drug to circulate and where it goes in the body. With the Hurel system, drugs are exposed to a more realistic physiological environment, thus providing higher and more accurate informational content than traditional in vitro assays.
Another way of predicting what effects a drug may have in the human body is to find out which genes it affects and in what way. A Cambridge company, Simugen, uses the latest cell and DNA-based methods combined with advanced computer programs to predict how toxic a drug will be. Their methods have been able to detect hazards to the human liver that were missed by animal tests.
What we do is use mathematical models of how the genes are actually changing to predict those dose-response relationships. So it's a very, very exciting technology because we're addressing several things. First of all, we're giving you this richness of information that current methods can't give you. We can predict toxicity, we can tell you the level at which it's going to be toxic, we can give you some indication of what you could be doing to make it less toxic, and we're doing it in a way that is very cheap, very fast. It takes 24 hours to do a cell culture. So this is something you can use right at the beginning of, for example, a drug development pipeline as a company, which will save an enormous amount of money and decrease the reliance on animal testing.
A further way of predicting what effects a new drug may have on the human body is by computer modelling.
The image you're looking at may appear to be a film of a living human heart, but it isn't. It's a highly sophisticated computer program using tens of millions of separate equations, combining graphics and mathematics to create one of the first of a new generation of simulations called virtual organs.
My involvement in modelling the heart and other organs began over 46 years ago when I was working in London. I was the first person to build a computer model of the activity of individual heart cells as they were beating. And that was based on experimental information that we obtained on the nature of the processes occurring in those cardiac cells. Now, those models were very simple and they ran on very, very simple computers. Nowadays, 40-odd years later, we have masses of more information, thousands of laboratories all over the world accumulating experimental information. The models have become very much more sophisticated, and most importantly, they've been incorporated into models of the whole organ so that you can actually see the organ itself beating away.
The virtual heart allows doctors to study the effects of a heart attack in controlled circumstances without endangering a real patient. And unlike real life, in which a heart attack can take just a few seconds, the progress of the attack in the virtual heart can be slowed down, speeded up, and repeated at will. Virtual organs such as this one allow researchers to develop and test new drugs, faster, more confidently, and at less cost.
One of the best ways of finding out how a new drug will behave in the human body is to give it to volunteers at a minute, safe dose. Then, by using incredibly sensitive measuring devices, scientists can monitor where the drug goes and how it is broken down in the human system. Microdosing is an exciting new breakthrough in drug testing. It uses machines so sensitive that they could detect a litre of liquid after it had been diluted in all the oceans of the world.
Now, why is microdosing of interest? Well, it's of interest because it allows us to get human metabolism information much earlier, much more safely, and with minimal use of animals. Now, the great attraction of microdosing from a drug development point of view is that you can take your drugs into humans with much reduced costs and much reduced time compared to classical, traditional so-called phase 1 methods. So typically, to take a molecule into man using classical phase 1 methods might cost as much as £3 to £5 million and take anywhere between 12 and 18 months. Whereas using the microdosing approach, we can do the same type of study at much less cost, typically around about £250,000, and get the results in 4 to 6 months.
These new technologies enable drugs to be developed and tested specifically for humans. Yet there is still a vocal strand of opinion which insists that new treatments will only come from research involving animals, and that it would be unethical to expose people to drugs without first showing that they are safe in animals. But this view is increasingly questioned by members of the medical profession.
We commissioned one of the world's largest market research companies to conduct a survey of 500 GPs across the UK. The survey found that 83% of GPs share our desire to see the medical value of animal tests evaluated scientifically. That's an extraordinary level of concern.
Many politicians are concerned about animal experimentation, but believe there's a dilemma between protecting animal welfare and advancing medicine. Yet there is growing evidence that favouring the state-of-the-art methods we've seen in this film would benefit medical progress as well as laboratory animals.
A lot of doctors and scientists are now challenging the scientific value of the use of animal testing for human purposes. And these arguments have to be taken seriously. 250 members of Parliament have signed a motion in the House of Commons calling for a government inquiry. And what is really required is a proper scientific evaluation of the methods that are used to decide, or make it possible for us to decide, what is the value of animal testing. And the government should now, in my opinion, make this a priority, because if they did that, then this argument could be settled in the best possible way for the benefit of everybody concerned.